welcome to everyone. Welcome particularly to the team members who are present and to the presenters who will identify themselves shortly. So thank you for coming. Thank you also for the preparation that you have done for this meeting. It's much appreciated. I think most of those, if not all, on the phone know of the program stewardship and strategy, the PSSC, and we're operating today in stewardship mode. We have a responsibility for reviewing periodically all of the projects that are ongoing within HESI and the technical committees, and essentially the purpose of the review in a nutshell is to ensure that there is a well-defined plan, that the group knows what it's doing, there's a commitment to productivity and evidence of achievement in what they've already done, and that there are clearly set objectives for the period which they're petitioning for continuation of the committee, and essentially thereby ensuring the overall quality of the work products of the HESI organization, and as a component of that, ensuring that what the group is doing is aligned with the mission and goals. So that's essentially what this is about. And before we move forward to the business, I would like to just deal with one or two formalities. Jennifer, I take it no one of the PSSC, no one on the PSSC who's participating has declared a conflict of interest?
Nope, no one has declared one.
And can we just check that for those of you who are voting members of the PSSC and who are on the phone, if anyone does have a conflict of interest, then if you say so, the most obvious conflict of interest would be a direct involvement with the committee under review.
And that would be me, Ernie. This is Scott. You'll hear me all the way through, and I assume at the point that it goes into deliberation, I will also step off.
That's correct, Scott. Yeah, thank you for that. Yes, as the committee co-chair, and I presume at least one of the presenters, yeah, at the point where we go into private session, closed session, then you would leave with the other members who are not conflicted voting members of PSSC. So you lead me nicely into that. The procedure, the conduct of the business will be, we'll listen to the presentation. You will have roughly 45 minutes for that to allow time for discussion. I would ask everybody to be respectful of listening to the presentation, go on mute, good practice for teleconference, and keep all questions to the end, unless there's something absolutely pressing or you can't hear or some difficulty. But otherwise, I think we will wait until the end, take questions, and in the first instance, I'll take questions from the members of PSSC who are present. So we'll have an open discussion. Everybody's welcome to stay for that. And at the end of that, if there is sufficient from those who are not members of PSSC, and then at that point, depending on time, we need to allow time for the closed session. Everyone will be asked to leave the call, except the PSSC members and HESI staff. Did somebody have a comment to make? Okay, so that's how we'll conduct the business. I don't think I've anything else to add. I think we have had some other people join while I've been speaking. I think yes. We can go ahead, Jen, or do you want to check if we are now quoried for the PSSC?
Yes, I believe Angelo has arrived, and so that should
exactly.
Start.
Very good, thank you, Angelo. So PSSC is quoried. We may have others. I know there are one or two, at least, others who were expected to attend, but as long as we're quoried, that's all that matters. So, Scott, are you going to open the presentation, or maybe you're doing it?
Yes.
So I'll invite you to talk us through the presentation, and as I say, if everyone can go on mute while you present, that would be very helpful. Thanks, Scott.
Great. So I'll hopefully keep Citrix working properly through this. If we get an interruption, we'll get it squared up straight away. This is actually the third review of this committee. It was originally formed in 2007 at the beautiful meeting in Cancun. And this is a committee whose primary goal is to work with scientists to develop a sound basis for alternative test methods to address 3R needs across the globe. It's acted as a very unique forum to coordinate a lot of the debate and emerging best practices in this area, and it's largely been unchanged in its mission for the last 5 years or so. However, what's really kind of interesting to us, for those who are deeply into this, animal alternatives was kind of new on the scenes for environmental in 2007. Today it's mainstream, and we see that more and more of the work that this committee does has broad technical bases for environmental risk assessment in general. And that'll become clear as we work through the presentation. Why do we have this committee? Fundamentally, it involves pressure on animal testing continues to influence both testing itself and testing strategies, and concomitantly environmental risk assessment practices. This can be now seen in legislation throughout the globe. Animal alternatives in both human and environmental science are specifically mentioned in the TSCA reform bill. Of course, we see broad implications in REACH and the Cosmetics Directive, of course, in Europe. And environmental tests in the pie chart to the right of this present screen, environmental vertebrate tests account for about 25% of undeclared testing proposals that have been submitted to the European Chemicals Agency, meaning that it is very clear that there is a need for this work. The mechanistic emphasis that's occurred in environmental risk assessment in the past few years has also become pretty central in this. And the HESI committee acts as one of the most well-coordinated forums to have this kind of deliberation. The makeup of the committee, it's not as large as some of our other committees in HESI, but it is exceptionally active, and it'll be seen throughout the presentation how this really looks. There are about 35 participating organizations that are active, and I would say that there are in excess of 100 individuals across those 35 organizations. It's interestingly nearly completely balanced with respect to academia, industry, and government participation, and we even have a small number of NGOs that routinely are involved with us, including Humane Society International and NC3Rs in the UK. Our participation is also very nicely divided amongst North America, Europe, a little less in Asia, and then pan-global groups. But while these pie charts look the same, they are not the same people and organizations that are represented both. That's just how the numbers kind of work out. And I would also like to note here, and I'll probably note again at the appropriate point, we have a new industry participant that began in 2015, and we welcome Dow Chemical to the active participating list of this group. These are the principal active participating organizations. Those that are in red are represented on the steering team. It's an exceptionally strong group of organizations to work with, and we have been blessed by working with some of the best that the environmental sciences have to offer. I couldn't begin to put names all down here because we have so many individuals that are multiple under certain categories, but you get an idea of our breadth of participation. The last 3 years has been pretty satisfying, I would say, as chair. I'll give Michelle a big shout out here. One of our seminal papers from our first workshop hit its 100th citation this last year. In only 5 years, it's still getting cited somewhere between 25 to 30 annually. There are 1,200 reads in ResearchGate. It's turned out to be one of our most influential products, and this goes back to the original work of the committee on fish embryo toxicity testing. A new program that you'll hear quite a bit about is our work on ecological thresholds of toxicologic concern. This is so hot that it was mentioned in the Joint Research Committee's recent publication on technical reports for 2015. The work is far from completed, but the anticipation of what we could do is very high, and the JRC found it fit to identify the work of this group in their annual report. We've got a lot of outreach activities. Admittedly, a relatively small group, 5 publications, 15 presentations from the last 3 years. A number of seminars and a new monthly seminar series beginning in 2016. The group is highly recognized at OECD for its programmatic contributions. We'll talk a little bit about this. One of its most important has been the role that the technical committee played in the formation of the OECD 236 test guideline on fish embryo testing, a very important activity that came to fruition in 2013 with the establishment of the International Test Guideline. The test itself is now present in almost every major contract lab that does work in environmental testing for regulatory compliance. Probably a legacy contribution that we will continue to keep an eye on for some time as it generates interest, questions, and is part of testing strategies that remain in development. In research, we had the authoring of a CEPIC long-range research RFP on adverse outcome pathways in fish chronic toxicity testing. I'll talk about that a little bit. Some work on effluent toxicity assessment, which was pure research. Some very interesting things going on with EcoTTC, and also modeling of apical endpoints in fish chronic testing. So this is de novo research, not sort of review or anything like that. This is the real deal. We've had expert meetings, several over the last 3 years, an AOP, Adverse Outcome Pathway Expert meeting in Duluth, Minnesota at US EPA. We've had 2 EcoTTC meetings in Washington, D.C., and a very big workshop coming up early next year on effluent toxicity and animal alternatives in effluent assessment. In terms of citations, something to make note of, our 2015 papers already have 150 downloads in the first 5 months that they were available. Committee publications on average are 28.1 citations per publication in Scopus. 42 different co-authors since 2010. Obviously, we've got a lot of reach going on here. In 2012, this was our scope of work for the animal alternatives and environmental risk assessment. We had work on fish embryo toxicity testing that led to this Cefic LRI proposal on adverse outcome pathways, and an interest in our group to maintain that. We'll talk about that in a moment, where that sits. There was work anticipated on whole effluent toxicity, or WET, as it is known in the US, or WET in the European context is whole effluent assessment, WEA, or direct toxicity assessment. We had programs of work on the OECD 210 fish early life stage test guideline, an update to it, and some subsequent work, and ongoing interest and programs in endocrine disruption and in vitro testing. 2 of these programs of work, I would say, have either come to fruition and we're done with, or they're kind of residual activities, and we won't talk about them too much. The Cefic LRI program was funded at 750,000 euros to a group in Belgium that is known as EcoTTC, and it has been highly successful. This would not have happened if it wouldn't have been for our group. But we don't feel it necessary to drive and be part of this at this point. We got the work off the ground through workshops and funding, and it is really productive in its own way, and it's gone off and done very nice things. So we've stepped away from the AOP work. Similarly, the fish embryo test, we have some residual involvement after the guideline was adopted, but it's no longer an active programmatic area for us at this point. So what is active? We've got 5 areas of work. Whole effluent toxicity and effluents, some interesting things going on with life stage terminology and developmental biology, some of the work on EcoTTCs, continuing work on the OECD 210 and the endocrine disruption chemicals and in vitro testing. And we'll walk through these in the science that is involved. We'll talk about what the programmatic area of work is, what have they accomplished, and what's the plan moving forward. In broad strokes, a lot of the work is developing of frameworks and appropriate models that can be used. So the EcoTTC has been largely to develop the framework and demonstrate principles of action. Under the OECD 210, we've got some development of some new models with new endpoint insights. For the endocrine disruption area, the point of emphasis are principles for use in vitro validation of new methods, and there's some significant work there. Life stage terminology is another area that we have picked up with OECD, and it involves harmonization of terminology across the dozen-plus fish test guidelines, which frankly are a mess. And lastly, in wet testing, we're identifying the roles for animal alternatives and involved in understanding what is the state of science here. The steering team, which is composed of a dozen individuals split across the sectors as well as geographic areas, oversees the 5 main buckets of work. And here I won't go through what the lists of individual groups here under each, but suffice to say that many of these, where there's a number following the organization, those will represent numbers of individuals, or in some instances, if it's a government group particularly, it might represent the number of departments or divisions involved. So again, a very broad reach of folks with lots of cross-sector involvement. So what is some of the work here? The work on life stage terminology is been something that's kind of bubbled at a low level for about the last 4 years. It grew out of our fish embryo test work, where developmental staging is a key component of executing the test. The test guidelines for OECD and US EPA and other jurisdictions have been built over decades. However, fish, which vary tremendously in life history and reproductive strategy, variously fall under different descriptions of their developmental biology. Add on top of that, there's an obfuscation of terminology across fisheries biology, ichthyology, ecotoxicology, and developmental biology. And in fact, even though we may all be talking fish, many of these groups rarely talk with each other. So what we discovered is that across the test guidelines, there is a great variation in terminology used. This picture of a developing young fish, one might call it a sac fry, which is a fisheries biology term. One might call it a hatchling, which is a lay term, but it means something different than a sac fry. It may be an eluthroembryo, which is an appropriate technical term, but not synonymous with sac fry. And in some instances, some may refer to it as a larva, which it is definitely not. And the test guidelines use every one of these, sometimes interchangeably, which results in confusion both for regulators as well as those who do tests. So this sub-team has worked on developing recommendations for harmonizing life stage terms across the myriad of test guidelines of EPA, ASTM, ISO, and OECD. And this would assist the accounting of animal use, as well as improving consistency and transparency of how developmental stages appear in adverse outcome pathway work. So OECD requested of this technical committee input and assistance to do this harmonization. HESI findings and recommendations on this were presented to the OECD Fish Drafting Group, which is a working body which exists under the Test Guidelines Program, the OECD Ecological Validation Management Group, and it's also been presented at several CTAC meetings. And we've proposed to resolve these by publishing the findings of the subcommittee's work, which includes an overview of species diversity in chronic fish testing, a formal review by OECD, and finally, adoption of a work plan to amend or harmonize test guidelines. And this last one, this is a big job when we get into test guideline revision. But our review in this area is going to provide a requisite roadmap to make that happen. In the area of ecological TTCs, this is an entirely new arena of research in environmental risk assessment. It's got very heavy involvement by the European Commission and US EPA, and this is because the regulators see great potential. It's really quite exciting. What this has turned into, interestingly though, is almost a charting of the environmental risk assessment process, something I might not have anticipated, or the group would not have anticipated in the beginning. To make this come to life, we've spent a significant amount of time developing a very large access database of individual test compounds, their chemical properties, taxonomic information on the organisms tested, and ecotoxicity information. This is hundreds of thousands of potential data points. Literally 1,500 different species tested, and somewhere between 4,000 and 10,000 chemicals worth of data. Access is our platform within which to work this, and we've begun to filter through the information using a series of process diagrams which chart out how mode of action assignment occurs, how functional groupings of chemicals can be thought of, how to include studies based on adherence or relevance to test guidelines, and PNEC derivation, or the final identification of the hazard concentration. And these lead to eventually the ecological thresholds of toxicologic concern. And this is an example of one for nonpolar narcotics, where the 5th percentile of the distribution of toxicity values, 309 different compounds, is charted. And we suspect that this is just the beginning of probing this process. So the EcoTTC, why are we doing this? It can be useful for compounds that are used at low volume. It provides a first cut guidance for new technology development. It could be used for groups of compounds for which quantitative structure-activity relationships are not available, like polymers. It could be a way to augment weight of evidence assessment. And through these various uses would reduce the need for animal tests, as well as other tests, actually. How do we see this working? This is a classic flow diagram for data development, where we're spending a significant amount of time on collecting information. The access database that we have developed is highly sought after already by other groups, and we are looking at partnerships beyond HESI to utilize the information once gathered. We have a process of harmonizing the information in terms of quality and architecture, characterization of the information by for both chemicals as well as the biology, a process to then derive predicted no effect concentrations, or PNECs. The DNL in human safety literature would be the equivalent, and then a whole host of data analyses to look at what the distributions of PNECs look like by structural group, by mode of action, by species tested, and from these predict the 5th percentile and confidence intervals for a given category. This is just in the early stages of development, but we've already got incredible levels of progress to report. The philosophical basis for this has been outlaid in a initial paper, which is just in print as of last week. The database structure is well on its way, and we have been working on the development of access experts within the group. Decision systems are well outlined for a variety of the items of concern. We've automated the PNEC derivation process in a PNEC calculator using statistical programs, and there are some statistical models that are in development that we are working to as well. And we've got initial evaluations. Does this have a reason for being? Does it work? And it sure looks like it has a good reason for being. The second paper, which is going to be a demonstration of the process for a chemical group, is in development and probably will be ready sometime second quarter of 2016. There is a whole host of work that is involved here to educate and recruit additional scientists, streamlining information, a whole bunch of papers that are planned, and we are moving to port the calculation systems and databases to a desktop environment from a server environment. The second another, I guess this is the third major group of activities, is in the modeling of hierarchical responses in fish chronic toxicity tests. And this is the OECD 210 work. In fish testing, apical endpoints in traditional tests are treated as independent endpoints, when in fact they are not. When fish die in a test, the surviving members embark on a different growth pattern than if all the organisms did survive. And in the end, we have survival metrics, a variety of growth metrics, and a confounding factor can even be, for some fish in testing situations, mortality will release the potential for fish to grow greater as opposed to grow less, even though survival has been impacted. Well, this is actually quite a complicated environment, and fish have indeterminate growth. That means they have a maximum size which is really a function of their food density, predation, and competition. Food availability and organism density clearly influence the maximum growth potential in ecotoxicity tests, and even influence the maximum growth potential in the real world. A famous exploration of this phenomenon is the identification of the Loch Ness Monster in Loch Ness, Scotland, and based on ecological principles, only one monster can be present in Loch Ness based on food availability alone. The project in this area was actually executed by funds as a research opportunity to evaluate opportunities to combine patterns of mortality and hatchability, which are dichotomous, yes, no, do I or don't I, and growth in terms of length and weight, which are continuous responses, into a single endpoint, because these two are really related. It drove out of our work on the OECD 210 chronic toxicity database, which we reviewed in the last committee review in 2012. Bayesian models have been explored using several data sets to see if this is actually possible, and Dr. Jin Jiang, Jim Orris, and John Baylor, all at University of Miami in Ohio, have explored these opportunities. And where we are is that sophisticated models are required to integrate these endpoints, both the temporality of them as well as exposure response. Data sets from the 210 review were shared to assess model flexibility and possibilities, and proof of principle has been achieved, and it can be done, and we're in the middle of now assessing the breadth of applicability. There will be a seminar to this topic to the technical committee in the first half of 2016. It's still somewhat in development, but we clearly see two publications to be written up, one in a statistics-oriented journal where you can decode the lines of math that follow there, and a second in an environmental toxicology journal, which will place this information sort of into less mathematical and less statistical terms, but more in executional terms for the OECD 210. We shall see if this has legs for regulators to work with. Another area of work has been alternatives to in vivo tests for endocrine disruption chemicals. This is particularly in the area of reference criteria. Considerable effort in the committee has been put into this over the last 4 or 5 years. Scholes et al. Published an exceptionally detailed review of in vitro tests and their use in endocrine disruption activity identification, and this is a natural outgrowth of that work. The hole that the group saw was that very little attention has been paid to establishing criteria for chemical choice in vitro validation studies. The validation studies that are done for in vivo assessments use a very, very small set of chemicals. So the opportunities there for really diving into this in terms of a chemical diversity type of way is not there. But in vitro tests, which are used as the prelude to those in vivo systems, this is where you can really make hay. And the question always with validation is sorting out what are truly negative and truly positive endocrine disrupting chemicals. And surprisingly, this sort of fundamental assessment of what these chemicals should be so that in vitro tests can build even on each other has not occurred. So the team has developed a set of reference criteria for these chemicals to propose for validating assays that allow researchers to better interpret results. And this is for new or unknown compounds. Very weak criteria for compound selection, I would argue, exists in present validation programs. And this has the unfortunate result of perhaps resulting in assay bias for researchers because you inadvertently relate observed effects through correlation, but not causation. This simple diagram here highlights the areas that the team has worked through, evaluating relevant chemical properties, ensuring agreement across assays for consistency, evaluating the tests and chemicals for effective scope, and then identifying chemicals that interact across multiple endocrine disruption pathways. And from this, a manuscript that builds on the Scholes et al. Work is in draft form, should be submittable in 2016, and then outlines experimental follow-ups for the greater environmental community to take up. The last area of work that I'm going to cover is animal alternatives and effluent testing. And you've heard us touch on this in past reviews, but this is the single largest use of fish in environmental testing globally. The statistic that is always in my mind is on an annual basis in the US alone, that amounts to 6 million fish that are used for effluent compliance assessment. Discharge compliance is regulated at the local, state, and provincial level, and effluents are regulated through a different regulatory authority from chemicals. This is not regulated federally, it's regulated more locally. And as a result, to deal with this is much more complex. Effluents are constantly changing, so testing is also periodically performed and required. It's increasing in developing geographies as these geographies catch up with chemical management and safety compliance at manufacturing. These areas are trying to implement tools to reflect the best science available, and this also includes less animal-intensive approaches. I think this is one of those areas where we have the highest goals involved, the highest goals of avoiding animal testing to begin with. And maybe we can build on things that we've learned along the way. The emphasis and trends in these areas tend to be on holistic assessments of chemistry, biological testing, as well as field monitoring. The trends are to use more chronic tests and fewer acutes, and to address particular modes of action like endocrine disrupting chemicals. So you can see there's even ties to our other work plan areas in this. So we have began in this area by exploring in an experimental way through a research program in Jim Orris's lab at Miami, and then also through his postdoc now at Texas Christian University in the U.S. We funded research to ascertain whether or not fish embryo tests could provide equivalent direction setting compared to what's known as the 7-day larval growth and survival test. This is an animal test, although a very early life stage test. We evaluated 2 different species, zebrafish and fathead minnows, and 2 types of effluents, and asked a variety of questions about those. And these culminated in 2 very nice manuscripts in environmental toxicology and chemistry. What did we learn? We learned fish embryo tests and larval growth and survival tests, so the embryonic stage, not non-protected assay in Europe, a bona fide animal test on the other hand with the LGS. They have unique advantages and disadvantages. They each have reason for being. In addition, zebrafish are better fish embryo test species, and fathead minnows are better larval growth and survival test species. So this was not where we hoped we'd end up, but this is the reality of the comparison. The differences and the strengths that they show in these assays reflect species-specific biology and development. We had hoped to use some non-destructive markers of growth, particularly for the larval growth and survival test, which utilizes length and weight. And one of the very interesting things that fish do is their ear bones, their otoliths, lay down rings like trees do. In early larval development, those rings are daily, and based on those ring patterns, you can calculate growth rates. It was too hard, said quickly. It would not be viable in any routine context. However, the team did look at some genomic expressions, and expression of genes associated with fish growth showed significant progress, and there's work to do here. This was quite exciting, and the team at Miami and TCU showed some really cool outcomes. This has actually led us to believe pretty firmly that animal alternative needs in effluent testing are going to require some significant shepherding. So the steering team decided that there was a need for a forward-looking workshop on the role of animal alternatives in effluents. It's been 18 months in planning and will occur in March at L'Oreal's research facilities in Paris. And the title is Concepts, Tools, and Strategies for Effluent Testing. The workshop goals are to review the state of science in effluent testing globally, explore the scope for reducing our reliance on animal tests, and identify and understand barriers to implementing new methods. It involves a very cool array of practitioners drawing from a global perspective. North America, Europe, Asia, Latin America, and Africa are all involved, 16 countries and 50 participants. This is going to be one very interesting meeting. The expected outcomes from the Effluent workshop, a series of manuscripts, case studies, evaluations of advantages and limitations of different assessment strategies, and essentially a state of science overview. We will probably also publish this as a separate standalone HESI report that will provide an overview of the entire activity. We want this on the shelf for others to use. This is also going to result, we believe, in development of an animal alternative in effluent's community of practice to connect practitioners, all to define a longer-term work program for the future. This is going to be probably, along with the ecological TTC, the major point of emphasis over the next coming 2-3 years. So taking all this and looking at in terms of timelines, 5 very active groups, all of whom are developing manuscripts, presenting externally, engaged in outreach with regulators, academicians, as well as the private sector. In the coming 6 months to a year, we believe that the hierarchical modeling and endocrine disruption reference chemicals probably will see a decline in activity, and we'll assess needs and capacities at that time, but parts of this will likely sunset as we really gear up in the EcoTTC and effluent assessment area. And there, as with our other areas of work in the past, workshop is going to be the launching pad for the next several years. With all of that, we propose that the committee be rechartered for 3 years. The committee itself is relevant to all the stakeholders. Our participation is energetic, it's motivated, it's quite passionate. We have vigorous debates on the phone about what's right, what's not right. The science is largely quite cutting edge, and the value for the participants is clear. Other organizations look at this one with envy, and we're pretty happy for that. So with that, I'm going to thank my committee co-chair, Thomas Brombeck. He sat silent through this in order to allow me to make the run without transitions needed and such. Thanks for taking your time out of the day